Okay, thank you for you guys. Thank you for Lynn's introduction, and thanks for you guys to come over to my complacent seminar. The topic of my study is shaping of human T cell phenotype and function by gut-derived certain fatty acids. We know, I haven't introduced myself, I'm sorry. My name is Yanhui. I'm a PhD student from Lynn's lab. And we know that certain fatty acids are organic fatty acids with less than 6 carbon atoms, and they are the bacterial fermentation products and act as a link between the microbiome and the immune system. The most common and most studied of them are acetate, propionate, and butyrate. It has been arrived much more interest in certain fatty acids. In humans, the microbiome analysis indicates that the bacterial species producing certain fatty acids and the concurrent certain fatty acids production are reduced in individuals with autoimmune disease and inflammatory disease. It is worth noting that the research of immunoregulatory function of certain fatty acids are based on mice. Those research involves the analysis of T cell phenotype and as well as the epigenomic modifications, gene expression, protein modifications, and cell metabolism. As for the T cell phenotype induced by certain fatty acids, it has been shown that the certain fatty acids facilitate the differentiation of regulatory T cells and induction of IL-10 expression. However, the evidence also shows that the certain fatty acids induce the inflammatory profiles of T cells, such as Th1 and Th17 responses, depending on the disease models and other factors. It is widely known that propionate and butyrate inhibit the histone deacetylases activity, HDAC activity. HDAC removes the acetylation of histone and is associated with gene silencing. Histone acetylation is increased by histone acetyltransferases, which is associated with the gene transcription. In addition to function as a HDAC inhibitor, certain fatty acids can also increase histone acetylation by being metabolized to acetyl-CoA and stimulating histone acetyltransferases. The transcriptomic study of the effect of certain fatty acids on T cells focuses on the specific gene expression. There is still a lack of the genome-wide expression study in both mice and human. The previous studies has demonstrated the effect of certain fatty acids on the expression of cytokine genes and the T cell lineage specific transcription factor genes expression. In addition to the histone acetylation, the acetylation of non-histone proteins have also been reported to be associated with the certain fatty acids induced T cell phenotype. For example, acetylation of non-histone protein S6 kinase, which is the downstream target molecule of mTOR signaling pathway, and the glycolytic enzyme GAPDH has been reported to be increased in response to acetate or propionate, revealing a role of the mTOR signaling pathway and the non-metabolic function of GAPDH in modifying T cell function. Before we have a look at the effect of certain fatty acids on T cell metabolism, it is worth noting that T cell function is intimately linked to cell metabolism. The key metabolic processes, glycolysis, fatty acid, and mitochondrial metabolism, are recognized as crucial players in T cell activation and differentiation. I would like to highlight that it has been reported that the intermediary metabolites or metabolic enzymes could regulate effective phenotype of T cells by performing non-metabolic functions. For example, GAPDH, the most abundantly expressed glycolytic enzyme, when it's not engaged into glycolysis, bind to the 3' UTR of interferon gamma mRNA, blocking its translation. Those findings indicate the complex relationship between T cell function and metabolism. It has been reported that in response to acetate, the production of acetyl-CoA was increased. As we all know that acetyl-CoA is the cornerstone of fatty acid metabolism. Certain fatty acids can be oxidized in mitochondria, followed by TCA cycle, and further oxidized by oxidative phosphorylation, OXPHOS, to generate ATP. For the fatty acid synthesis process in cytosol, the acetyl-CoA is firstly converted to citrate and transported to cytosol or nucleus, where it is converted back to acetyl-CoA for fatty acid synthesis or for the activation of histone acetyltransferases. However, those studies I have been talking about are based on mice study, and there is appreciation of importance to explore the effects of certain fatty acids in human T cells. So the aims of our study is to determine the effects of certain fatty acids on human T cell phenotype and function, and investigate the molecular mechanism by which certain fatty acids regulate T cell functions. Our hypothesis is that certain fatty acids modify the phenotype, metabolism, and function of human T cells. To achieve the aims of our study, we just sought naive CD4 and CD8 T cells from healthy adults PBMCs and activate them in vitro with anti-CD328 microbeads and exposed them to sodium acetate, sodium propionate, and sodium butyrate to investigate the induced T cell phenotype as well as the epigenomic modifications, transcriptomic and protein modifications, and metabolism. We include the flow cytometry, suppressive assay, bioplex, RNA immunoprecipitation, and chip qPCR, among others, in our study. Similar to the mice study, the first thing we look at is the phenotype induced by certain fatty acids in human T cells. We found that among those 3 certain fatty acids, acetate didn't show any effect on the cytokine expression. Propionate and butyrate significantly increased interferon gamma expression in CD4 T cells, and this phenotype was only observed when CD8 T cells were activated in the presence of IL-2. We all know that in response to activation, CD4 T cells produce higher amount of IL-2 than CD8 T cells. So to determine if IL-2 is required for the interferon gamma regulation by propionate and butyrate, we included IL-2 neutralizing antibody. We found that the induced interferon gamma expression by propionate and butyrate was abrogated by IL-2 neutralizing antibody. This supports the role of IL-2 in mediating the regulation of interferon gamma expression by certain fatty acids. Besides, we also found propionate and butyrate increased IL-10 expression. And also we found propionate and butyrate induced the proportion of T cells co-expressing interferon gamma and IL-10. It has been reported that the T cells co-expressing interferon gamma and IL-10 exhibit the regulatory capacities. We next aim to characterize those T cells. So by measuring the markers that have been reported to be associated with induced regulatory T cells, we found that the expression of CD25, FOXP3, TIGIT, CD137 were all overexpressed in T cells co-expressing interferon gamma and IL-10. So to further to determine the suppressive ability of certain fatty acid-treated cells, we performed the T cell suppression assay. In T cell suppression assay, we just co-culture the potentially suppressive T cells, in our case, butyrate-treated CD4 or CD8 T cells with conventional CFSE dye-labeled conventional T cells for 3 days, and quantify the number of divisions by CFSE dilution. We found that butyrate-treated T cells was highly efficient in suppressing the proliferation of conventional T cells and showed more suppressive ability than the cells prepared from the control conditions. To determine the role of IL-10 and interferon gamma in the suppressive ability of butyrate-treated T cells, we include the IL-10 receptor blocking antibody and the interferon gamma receptor blocking antibody in the co-culture system. The blockage of IL-10 and interferon gamma signaling in the co-culture system resulted in the reversed suppression induced by butyrate-treated CD4 T cells. Highlight the role of IL-10 and interferon gamma in assisting the suppressive ability of butyrate-treated T cells. So in summary, interferon gamma is induced by propionate and butyrate, and this induction is dependent on IL-2. Butyrate-induced interferon gamma and IL-2 co-expressing cells share the molecular characteristics associated with induced regulatory T cells. Butyrate-exposed T cells exhibit regulatory function, and IL-10 and interferon gamma contribute to the suppressor function of butyrate-treated T cells. So we next to explore the genome-wide expression changes induced by butyrate, we just sorted naive CD4 and CD8 from 3 healthy adults and exposed them to butyrate with or without activation and analyzed by RNA sequencing. We found that the first MDS plot shows the first source of variation is activation, followed by the cell types. The second MDS plot shows that butyrate treatment is shows how butyrate treatment constitutes the third source of variation, with 10% of the explained variance. This slide just shows the differentially expressed genes induced by between groups. Take CD4 T cells, for example. The pink represent the differentially expressed genes caused by butyrate treatment in activated CD4 T cells, and the green stands for the differentially expressed genes related to butyrate treatment in naive CD4s, and the overlap area this shows the up or down regulated genes by butyrate regardless of the activation status. This slide shows the butyrate, so the differentially expressed cytokine genes by butyrate. We found IL-10 and interferon gamma were all upregulated by butyrate, and the other IL-10 family members, they all upregulated. This slide shows the effects of butyrate on expression of transcription genes. Transcription factor genes involved in T cell differentiation. We found butyrate increased expression of TBX21, which is associated with Th1, and FOXP3, which is associated with induced regulatory T cells, and our gamma T is associated with Th17 cells. They are induced by butyrate. So, in a brief summary, butyrate induced significant changes in T cell cytokine gene profiles, in addition to interferon gamma and IL-10. Butyrate induced transcription factor genes relevant in T cell lineage differentiation. We next validate the expression of interferon gamma and IL-10 by doing the bioplex, qPCR, and flow cytometry, and confirmed the induction of interferon gamma and the agreement in the increase at the mRNA level and protein level. However, interferon gamma expression shows a greater inter-individual variance at the mRNA level, but not at the protein level. Which leads us to believe that there might be the post-transcriptional regulation happen to interferon gamma. As I just mentioned in the background, GAPDH, glycolytic enzyme GAPDH, perform non-metabolic functions to regulate interferon gamma expression, and it has been reported that nearly all the metabolic enzymes can be acetylated. It reminds us, is GAPDH acetylated in response to certain fatty acids? So then we just used the anti-acetyl lysin-specific antibody to probe the cell lysis from certain fatty acids exposed CD4 T cells, and found that the prominent band detected is at GAPDH site. And the propionate and butyrate exposed CD4 T cells showed higher acetylation level of GAPDH. We then precipitate GAPDH protein and blot with GAPDH, or precipitate acetyl lysin protein blot with GAPDH, or precipitate GAPDH and blot with acetyl lysin, and confirmed the increased acetylation level of GAPDH in butyrate-treated CD4 T cells. So we just wonder, is acetylated GAPDH involved in the post-transcriptional regulation of interferon gamma and the other cytokines? to determine if acetylation of GAPDH alter its binding with the cytokine mRNAs, which is perform the RNA immunoprecipitation and precipitate GAPDH protein and its cross-linked mRNA and perform the qPCR. We found 8 times increase of interferon gamma transcripts bound to GAPDH protein in butyrate-treated CD4 T cells. And the other cytokines were not, the other cytokines including IL-10, IL-4 were not detected, indicating a specific role of GAPDH in regulating interferon gamma expression. And this enhanced binding was associated with increased acetylation level of GAPDH in butyrate-treated CD4 T cells. We next designed the primers for 3' body and 5' of interferon gamma mRNA, and found that only primers for 3' of interferon gamma could detect the interferon gamma expression with the pulled down GAPDH protein. It suggests that the acetylated GAPDH protein binds to 3' end of interferon gamma mRNA. So we next to determine if acetylation of GAPDH regulates interferon gamma expression post-transcriptionally by increasing the mRNA stability. We performed the actinomycin D cheese experiment. Actinomycin D just inhibits the RNA polymerase II to block the transcription and therefore allows measurement of mRNA degradation. We found that in butyrate-treated CD4 T cells, interferon gamma mRNA showed higher stability. So this would be our working model. The acetylated GAPDH protein just preferentially bound to 3' of interferon gamma mRNA and protected from degradation, therefore promoting interferon gamma expression. So we just wonder if we just if we transfected the cells, T cells, with acetylated GAPDH peptide, can it mimic the effect of butyrate, the induced T cell phenotype? We just select one lysine residue from the foremost studied lysine in human GAPDH, which selected 254. Acetylation at 254 has been reported to promote glycolysis. So we just developed 3 different types of GAPDH peptides. One is GAPDH peptides containing acetylated lysine at 254, and a Y-type GAPDH peptide, and a GAPDH peptide containing a mutation at lysine 254. We found that the transfection of GAPDH peptide containing the acetylated lysine at 254 is associated in CD4 T cells is associated with increased interferon gamma expression, both as opposed to the other 2 peptides. And all those 3 peptides did not alter the expression of IL-10, just confirming again the specific role of IL-2 of GAPDH in regulating interferon gamma expression. So in summary, in butyrate-exposed CD4 T cells, in association with increased expression of interferon gamma, acetylation of GAPDH is increased. Association of acetylated GAPDH with interferon gamma 3' mRNA is increased, and interferon gamma mRNA stability is increased. The effect of acetylated GAPDH to increase interferon gamma expression is mimicked by GAPDH peptide acetylated at lysine 254. What we have found that regulation of interferon gamma expression is dependent on IL-2, and acetylated GAPDH is increased. Acetylation of GAPDH is increased in response to butyrate. It has been reported that IL-2 plays a role in promoting glycolysis, and acetylation of GAPDH is associated with a higher glycolytic activity. So we just wonder, could certain fatty acids also induce interferon gamma expression by promoting glycolysis? IL-2 has been reported to promote the glucose entry into glycolysis. This finding is validated in our study. We found the CD8 T cells with lower glucose, with lower IL-2 production, showed a lower glucose uptake and lactate production than CD4 T cells. When IL-2 was added, CD8 T cells showed the glucose uptake and lactate production were all increased in CD8 T cells, just to confirm the IL-2 promoting glycolysis. However, the glucose uptake was not increased in response to certain fatty acids, and this finding was confirmed by measurement of lactate production and the the measurement of extracellular acidification rate by Seahawks assay. We found that lactate production and ECA were all not altered by butyrate. The gene enrichment analysis also showed that glycolysis was not changed by butyrate. So we found glycolysis is not altered in response to butyrate, and glycolysis is required for the induced T cell phenotype. So we just included 2DG to block glycolysis, and found that the induction of interferon gamma expression by propionate and butyrate was reversed by 2DG. When we culture the cells in the absence of glucose, we found the ability of propionate and butyrate to increase interferon gamma expression was decreased. So in summary, IL-2 increase glycolysis. Certain fatty acids do not directly alter glycolysis. Blocking glycolysis reverses the ability of certain fatty acids to increase interferon gamma. So the glycolysis is necessary, but not a sufficient requirement for interferon gamma regulation by certain fatty acids. Then we just certain fatty acids do not alter glycolysis, but do they have other metabolic roles? Then we just look again. First of all, we just look again at the gene enrichment analysis and found that within the mitochondrial metabolism, the OX1 is very significantly decreased by butyrate. To determine the effect of butyrate on mitochondrial function, we measure the mitochondrial mass and the mitochondrial membrane potential, and found that butyrate decreased mitochondrial mass and membrane potential, confirmed the decreased OX1. Then we just look at to investigate the other steps of certain fatty acids pathways. We look at the expression of CPT1, and we found which is a enzyme critical for fatty acid oxidation. We found propionate and butyrate increased CPT1 expression based on the Western blot and the RNA sequencing data. For the other, and in just indicating that the butyrate or the certain fatty acids, they can be transported into mitochondria for fatty acid oxidation. And for the other enzymes that are involved in fatty acid synthesis, we looked at the fatty acid synthesis expression and found that certain fatty acids didn't significantly change its expression or even decreased it. To test the involvement of fatty acid oxidation, we included the automoxer, the inhibitor of CPT1, and found that the increased interferon gamma expression was reversed by automoxer. In other words, fatty acid oxidation is required for the induced T cell phenotype. So this would be our working model. We have found decreased OX1, increased expression of CPT1, and the involvement of fatty acid oxidation. Evidence that certain fatty acids oxidized in mitochondria, but not further oxidized by OX1. They might be converted to citrate and transported to cytosol or nucleus and converted back to acetyl-CoA for acetylation of histone acetyl transferases. So further experiments are required to measure the concentration of acetyl-CoA and citrate, and also the activity of histone acetyl transferases. In summary, in the presence of butyrate, mitochondrial OX1 in T cell is decreased. Expression of CPT1 also is increased. Blocking fatty acid oxidation reverses the induction of interferon gamma expression by propionate and butyrate. Butyrate has been also shown function as a histone deacetylases, its DAC inhibitor. We just stimulate the cells with a classic its DAC inhibitor, TSA, to say if TSA mimics the effect of butyrate. We surprisingly found that TSA increased IL-10 expression, but failed to increase interferon gamma expression, indicating that the underlying mechanism that might differ between butyrate-induced IL-10 and interferon gamma expression. To determine the relationship between histone acetylation and induced cytokines, we just had a look at the expression of an active histone marker H3K9 acetylation. We found we found increased acetylation level of H3K9 in butyrate-treated CD4 T cells. We next performed the cheap qPCR and found that the IL-10, but not interferon gamma, is enriched for H3K9 acetylation, indicating that H3K9 acetylation might involved in regulating IL-10 expression by butyrate. So in summary, acetate HDAC inhibition increases IL-10 expression. butyrate increased acetylation at H3K9 at the IL-10 locus. So this slide it summarizes the multiple mechanisms of butyrate effect on human T cells in our study. We have found that butyrate increased the expression of interferon gamma and IL-10, and also induced the proportion of T cells co-expressing interferon gamma and IL-10. However, the underlying mechanism might differ between those two cytokines induced by butyrate. Our results indicate that the increased H3K9 acetylation by butyrate is enriched at IL-10 locus, but not interferon gamma. For interferon gamma expression, we found increased acetylation level of non-histone protein GAPDH, which might preferentially bind to 3' UTR of interferon gamma and protect it from degradation. Two mechanisms has been characterized to increase the acetylation. We include from our results, just in our study, we just include a classic HDAC inhibitor, TSA, but TSA only increase interferon gamma, IL-10 expression, but failed to increase interferon gamma expression. Acetyl-CoA is the cofactor for activation of histone acetyltransferases. Acetyl-CoA involves the metabolism of butyrate. We speculate that butyrate oxidized in mitochondria and transported out of mitochondria in the form of citrate and will be converted back to acetyl-CoA in cytosol or nucleus for the choose acetyl-CoA and for the activation of histone acetyltransferases. And this idea was supported by our finding that OX1 was decreased and CPT1 expression was increased, and the involvement and fatty acid oxidation is required. In the next, we are going to measure the concentration of acetyl-CoA citrate and also the activity of histone acetyltransferases. Our transcriptomic study also shows the alteration of transcription factors induced by butyrate, but how it needs to further investigate how butyrate epigenetically regulate transcription factor expression, and how they will impact the expression of cytokines, and which of the other histone and non-histone proteins would be changed, and how they will modify the T cell induced phenotype. So in conclusion, certain fatty acids, especially butyrate and propionate, alter the phenotype and function of T cells. They might induce the interferon gamma and IL-10 expression by different mechanisms, just as I mentioned in the last slides. The function of GAPDH to regulate interferon gamma expression, and the histone marks at the promoter of IL-10 locus. And they also induce the suppressor function. Those effects of certain fatty acids model their functions locally in the clone, where certain fatty acids and IL-10 co-expressing regulatory T cells have been identified in mice. Furthermore, we just also collaborate with the Royal Melbourne Hospital for a double-blinded clinical study. The participants were advised to take control or butyrate-containing diet every day consecutively for 6 months. And at the end of the study, we took their serum and PBMCs for metabolomics and phenotyping. We found that the donor who had increased level of butyrate in serum is associated with increased proportion of interferon gamma positive T cells, CD4 T cells. This is consistent with our study. It's just the preliminary data. We are still working on it. So in the next, we are going to identify the lysine sites acetylated by certain fatty acids in GAPDH protein, and we are going to determine the effect of butyrate on the acetyl-CoA and citrate concentrations and the activation of histone acetyl transferases. We are going to determine the effect of certain fatty acids on epigenetic modifications, including histone acetylation at interferon gamma, IL-10 locus in T cells. And we are going to confirm the effect of butyrate in vivo and demonstrate the presence of interferon gamma and IL-10 co-expressing T cells in human clonic laminaria propria as reported in mice. Okay, then just I would like to, at last, I would like to express my sincere appreciation to my supervisors, Len, Yuxia, Nayara, and Tim, and thanks to Len for his continuous support for my PhD study, and for his patience, for his motivation, his immense knowledge, and his guidance just helped me all the time of my research. Thank you so much. And thanks to Yuxia, she has left, and she has moved back to China and is now working in Guangzhou. And she's very smart. And I think I'm very happy. I'm very lucky to have her as my supervisor, and she came up with this project and contributed stimulating ideas and encouragement. Then Nayara came and take over the responsibility to supervise me. She's super responsible, considerate, and thoughtful, and she just helped me coordinate my project and widen my research from various perspectives. And thanks to Tim, and he just gave me a lot of useful suggestions and advice on epigenetic study. And my thanks still goes to my PhD committee, Marnie, Axel, Malini. Thank you for you to agreeing to be my PhD committee. And I would like to express my appreciation to everyone in Harrison Lab, Gaetano. She just gave me a lot of instructions on the experiments, and he's an expert in doing RNA sequencing. And Asta, she conducted the bioplex for me and prepared the samples for the mass spectrometry. And she instructed me on doing Western blot and immunoprecipitation. I think the first two people I would, if I have any problems in the lab, the first two people I will speak to must be Gaetano and Asta. And John, Delsin, Katrina, Natalie, Alana, thank you for you guys, for your advice, for your patience, for your understanding. Your advice on my project you raised on the lab meeting are very, very helpful. And thank you for everyone in Harrison Lab. You guys create a very wonderful and warm working environment. And my thanks also goes to Michael Chopping. He helped me doing the cheap qPCR, and Maria from Tim's lab for her generosity, for sharing her antibody, and everyone in the FACS lab, they always there when I have any sorting problems, and also thanks to Brooke Flanders from the Royal Melbourne Hospital for doing the clinical study, and James from Melbourne University to help me do the seahorse experiment, and Tim and Tom, he just provided the reagents for doing mitochondrial function. And Jianguo, thank you very much for your advice on doing immunoprecipitation and analyzing the results, and the stuff from Bio21 for doing metabolomics, and the division coordinators, Natalie and Shu Hardy. Thanks for your patience, for your understanding, for your concern, and organize everything, organize the completion seminar and confirmation seminar each year for me. And I also would like to express my thanks to my grandparents, my parents, my brothers and sisters for their sympathetic ear, and I know they always stand by me. And they are also my friends, Li Yan, Qiao, Joey, and Jia Zhen. They are not here, but the happy times I have out of the lab in Australia is with them. And I also express my appreciation for the scholarship provided by Melbourne University and Weihai, the very excellent place provided by Weihai. Without all these things, everything cannot happen to me. And thank you for you guys for coming to my completion seminar. Thank you.
I don't move.
Yeah.
Not quite over yet. Despite the acknowledgement slide, you've left a few minutes for questions. She's be very happy to take questions.
I want to make a stab at it, just have a guess. What proportion of your effect do you think is due to the GAPDH acetylation? So you have thousands of genes changing, but obviously this one gene could explain the effect of interferon gamma. So do you think that's most of the result, or do you think that those other many genes that are changing are responsible for it?
I think, thank you for your question. And we have been thinking about this problem, and Nayara also just raised this question before. I think we just we are still working on it, try to know the proportions that has been isolated that plays a role in regulating the cytokine expression. Yeah.
Just wondering, is there a difference between propionate and butyrate at the molecular level? Propionate stimulate the HDACs as well as butyrate, or do they both do GAPDH? Is there a difference between propionate and butyrate?
Yeah, I think as far as I determined, I read a paper, then they just in mice, they found no effect for, they found the propionate showed no effect, but butyrate has showed effect. But in my experiments, as for the induced T-cell phenotype, propionate and butyrate, they shared the same T-cell phenotype. I didn't measure we haven't measured the HDAC activity yet. So we have no idea whether propionate and butyrate, they all inhibit HDACs to increase histone or non-histone acetylation. I just include the butyrate, but for the acetylation of GAPDH, we found propionate and butyrate, they all increase the acetylation level. We just assume propionate and butyrate might have the same mechanism to regulate the cytokine expression, but we haven't proved it yet.
So in the diet, I guess the short-chain fatty acids are thought to be because of bacteria in the gut digesting fiber. What sort of serum levels do you get of butyrate and propionate? How do they match with your in vitro concentrations that you use?
Yeah, that's the. I have to admit that we used pretty high, but still less than the reported level in mice. I mean, I use 1 molar. 1 molar for propionate and 0.5 molar for butyrate in my in vitro in vitro experiments, but still higher than the physical level in human body.
Blood.
Yeah, in blood, yes.
And so do you see it as something that's sort of globally changing the metabolic pathways, or do you see it as potentially having some sort of a receptor that is altering the function of one protein that's having lots of follow-on effects?
We try to look. We tried. Yeah, we have tried to look at the expression of G receptor, G protein coupled receptors that have been reported to mediate the effects of certain fatty acids. But we found that the very most studied GPR 41, 43, and 109A, they are all very lowly expressed in human T cells. They are undetectable from our RNA sequencing data. So we just think maybe there are other transporters expressed in T cells that mediate the effect, or just the transport certain fatty acids into the cells and for some cellular processes. Yeah.
But as you said in your last conclusion, this is a model for what's happening in the colon, where the concentrations are that high or higher. So I don't think, well.
Yeah, we just think we are using the T cells from the blood to modeling what happens what might happen in its production site, the colon. Yeah.
So you showed that you can get a similar effect on increased interferon gamma production with GAPDH acetylated peptides.
Yes.
How do you imagine that works? Do you think that a small peptide is recapitulating the binding and mRNA regulation effect of the intact GAPDH protein?
Could you please repeat again?
Well, if I read your slide correctly, you showed that acetylated GAPDH peptides.
Yes.
One peptide in specific could reproduce the effect of GAPDH acetylation. So if GAPDH is acetylated and then binds and regulates an mRNA, how do you imagine that a small peptide does the same thing? What's the mechanism of mRNA regulation by GAPDH?
So I think it's a good question.
I don't expect that you necessarily know the answer. I'm curious to see what you think.
It's a great answer. I think that's what you do think, right? That the peptide, that was the aim of the experiment, to see if you could focus on one region of GAPDH to mimic the function of the whole protein. Now, where that's happening, muscle waits for more RNA, experiments and competition and so forth, but they're in the process. I think that was the idea, right? That it would mimic GAPDH. You try and find which lysine in GAPDH. The other peptides are there. We just haven't, you haven't tested them all yet. You hit on one that did mimic first time. And it's actually, if you look at the structure of GAPDH, it's not only essential for glycolysis, but it's on the outside of the structure, very, very accessible.
How long is it?
So,
How long is it?
Well, I don't remember. Maybe 10. But amino acids 10 or I don't remember, but yeah.
Yeah, so thinking from a structural point of view, I think that's surprising but very interesting. And you showed two other peptides that didn't have the same effect, even though they were acetylated on different lysine. Is that correct?
Yeah, I just include another 2 control peptides. One is just the wild type, just that fragment, and the other one is with a mutation at lysine 254. Yeah, but they didn't show any effect. Actually, I don't want to make things conflict. It's sometimes for me hard to explain.
you did a good job. Great job.
Thank you.
Thank you. It's all over for now.
Yeah, thanks.